Good morning and welcome to second New Jersey Metastatic Breast Cancer Conference. I want to thank Suzanne and Komen, New Jersey for giving me and my lab member the opportunity to share with you some of the latest events we are doing in the lab regarding metastasis of breast cancer. So I'm going to start by sharing this slide showing that for cancer patients, when they are diagnosed with early stage cancer, including breast cancer and other cancers that you can see in this slide, the 5-year survival rate is quite good. For breast cancer, it's nearly 100% now. But for patients who are diagnosed with late stage, stage 4 metastatic breast cancer, as you can see in here in red line, the survival rate is much lower. And also, it doesn't really improve much in the last 10 years also. So the main challenge for decreasing the overall mortality for breast cancer is to find a better treatment for metastatic breast cancer. So when you look at the mechanics or the biology of cancer metastasis, these are the common diagram that we use in our research. It shows that for a tumor growing in the mammary gland, they have to overcome many different steps or obstacles in order to form metastatic lesions in other organs. The tumor cell have to invade through the nearby tissues into the blood vessels and then survive the turbulent circulation and attack by the immune system and eventually disseminate into distant organs such as the bone. And actually when they disseminate to distant organs, most of them do not grow up to life-threatening metastasis. Many of them remain dormant for many years, but eventually some of them, a very small number of those so-called disseminated tumor cells, eventually wake up and start to grow and become life-threatening metastasis to patients. So this is a very challenging and actually very hard to overcome obstacle for tumor cells. So for cancer cell to metastasize, not only they acquire properties to grow as tumors through, for example, activation of the so-called oncogene and the loss of the so-called tumor suppressors, they also has to acquire additional properties, the properties that mostly allow them to survive in stressful situations, be able to defend themselves from the immune system attack, be able to change metabolism, be able to also deal with, for example, a lot of proteins that are not made properly in the cell. So most normal cells cannot deal with this type of pressure and they cannot survive. But cancer cell, especially metastatic cancer cell, acquire those abilities through mutations or elevating the activity of certain proteins that allow them to survive those high pressure much better. And in the lab, we call them the so-called metastasis fitness genes that increase the competency of the tumor cell to survive the stress. So in terms of therapeutics, a lot of research in developing cancer therapeutic has been focusing on the so-called early drivers of oncogenesis. For example, I list some of them here: BRAF, KRAS. These are some of the common oncogenes, including in breast cancer, HER2. Those are common oncogenes that we heard about, and they have been major targets of development of therapeutics. But there's another area of therapeutic that has not been a focus in the past, but has becoming more and more important in terms of anti-metastasis therapeutics, which is in the later half of the so-called metastasis competence genes. I want you to maybe put this more complicated graphic into a more easily understood image. Just consider metastatic cancer cell as a pressure cooker. You know, it may look like very hard and very difficult to crack, but the cell is also under a lot of pressure. They are under high pressure, just like a pressure cooker. And if you can find a weak spot in that pressure cooker and puncture it, you could, the cooker will explode, and in the analysis to cancer cell, they will die. And so the challenge is to find those weak spots that we can attack and disable the defense of those metastatic cancer cells for different kind of stress. So to summarize, some of the focus of the lab, what we have been working on in the past few years, and you are going to see some examples presented by my lab members, Nicole and Mark, in those areas. So our underlying hypothesis for our research is that cancer progression and metastasis create a lot of stress that select for populations of cancer cell with enhanced fitness and greater metastatic potential. And those cancer fitness genes are critically important for the cancer cell, but they're not the so-called traditional oncogenes. And additionally, if we target such cancer fitness gene, we could cause potentially the regression of cancer. And also, such treatment might have relatively little toxicity in healthy tissues because normal tissues are not under the same kind of stress as cancer cell, and they are less critically dependent on those cancer fitness gene. And actually, in the field of cancer research and treatment of breast cancer, there have been some success in terms of targeting those so-called cancer fitness gene, including some of you probably heard about the so-called PARP inhibitors that target triple negative breast cancer that have deficiency in this tumor suppressor called BRCA1. And also the recent advance of immunotherapy is also try to target the weak spot of cancer cell and make them susceptible to immune cell attack. And I'm going to show you some of the examples that we have been making progress in recent years, and hopefully moving from the basic research in the lab into potentially new therapeutics in the clinic. So one of the studies that we have been working on in the past 15 years is a gene called MTDH metadherin, which we discovered back in 2009, showing that it's a critically important breast cancer gene that is hyperactive in cancer patients, breast cancer patients who are prone to develop resistance to treatment, to develop metastasis, and likely to have poor outcome in treatment. So this gene promotes both chemo resistance and metastasis of breast cancer. And a few years later, we use a molecular biology technique called crystal structure analysis to show how this metadherin protein, which I show in yellow, this is a piece of it, binding to its partner in crime that promote cancer in green, and this protein is called SMD1. And you may find this difficult to understand what this means, but just imagine this green SMD1 protein as a bowling ball, and the oncogene MTDH in yellow is using two fingers. So in red in here, these are two so-called amino acids that allow the MTDH to bind to the SMD1 protein using those little holes. So in cyan color in the surface of the SMD1 protein. And this is a critical interaction that allow the two proteins to hug each other and perform the key functions to promote metastasis and chemo resistance. And if you are imaginative, you could imagine drugs that could block their interaction together by those compounds. If they can insert themselves into those holes, just like you insert stuff into the bowling ball, the holes of the bowling ball, you will no longer be able to use your finger to hold on to the bowling ball. And so this is exactly what we try to do in the lab using some of the chemistry approach to synthesize molecules that do exactly that. And they have shown promise in treating metastatic breast cancer in animal models, and we are hoping to follow development in trials in the patients. Another example is in the treatment of bone metastasis, and this is again is a study that was completed in the lab a few years ago, in which we found that for tumor cell to grow successfully in bone, they need the help of the normal bone cells, including the cells, so in pink color here, that we all have, normal people, healthy people have it because we need to rebuild our bone tissue every day. And the so-called osteoclasts, so in pink color here, resolve the bone and allow the brown color osteoblasts, the bone building cell, to build a new bone, just like every summer, some of the drive and the roadways get repaved by removing the old asphalt and then laying a new layer of asphalt on top of it. So this process is hijacked and twisted by tumor cell so that the osteoclast activity become hyperactive and resolve a lot of bones, and that cause fracture, bone pain, and other conditions like hypocalcemia in patients. And this tumor cell, so in blue color, achieve this by secreting proteins and factors that communicate with the bone cell. And one of the protein we discover is so-called JAG1. Again, this is a protein expressed in the surface of tumor cell and now the handshake between tumor cell and the stroma cell. The protein also play another role in the situation of chemotherapy, and it help protect the tumor cell from the toxic effect of chemotherapy and make the bone metastasis much more resistant and hard to treat using standard chemotherapy. So we collaborate with a drug company called Amgen in California to develop a neutralizing or monoclonal antibody against JAG1 protein. And again, we show in a mouse model that if you treat the mice with either chemotherapy or neutralizing antibody, you reduce bone metastasis burden in the mice, as showing those signals here, just like scans, PET scans in the patients, and they're using a different kind of signal to show the burden of bone metastasis. But interestingly, when you combine chemotherapy with JAG1 neutralizing antibody, we can almost completely eliminate bone metastasis burden. So this is showing promise of combining the traditional chemotherapy approach with more targeted treatments to increase the efficacy of bone metastasis treatment. So this is also showing promise, and we're hoping this eventually could benefit the patients who are suffering from bone metastasis. And the third area of research that Nicole is going to talk more is regarding the connection between normal stem cells in the breast versus the cancer stem cells in breast cancer. And it turns out that cancer cells do not try to do things completely new. They often hijack, just as you see in the bone metastasis, they hijack some of the normal activities of normal cell, but twist it in some ways to benefit the mimicking functions of the cancer cell. And some of the example we found is that, for example, the stem cells, they are also under certain stress from the surrounding, for example, immune cell. And the normal stem cell could prevent, becoming older, so-called senescence, or becoming less able to produce the mammary gland, losing the so-called stem cell activity. Such an effect could be caused by some of the immune cell producing inflammatory factors in the environment. And the same situation also happening in cancers. Cancer stem cell could become senescent or lose their activity by such factors. But this normal stem cell have a defense mechanism for that by producing a small microRNA that defend the aging function of those secreted proteins from immune cell. Such activity was again amplified by triple-negative breast cancer to allow the cancer stem cell to sustain their cancer stem cell activity, and that caused not only tumor growth but also metastasis. Again, this is relatively early stage in discovery, but hopefully such new insights from biology could help lead to new treatments for certain subtype of breast cancer, like triple-negative breast cancer. And in a few minutes, Mark also will show some of the interesting work that he's doing in the lab, showing again the interaction between the breast cancer cell with another kind of so-called surrounding stroma or microenvironment cell, normal tissue cell called blood vessel cell. And how this interaction between tumor cell and blood vessel cell in the bone facilitate the development of bone metastasis. So I'm not going to go into detail because Mark will also tell you the story, so I'm not going to steal the thunder from him. And finally, I want to end by saying cancer research, breast cancer research and development is a great team effort. The work I'm showing you are all done by very talented graduate students and postdocs in the lab. You see some of them in here. And you're going to hear more from Mark and Nicole, who are members of the lab. The research that we are doing is also possible because of the support of Komen advocates and a lot of fundraisers for the Komen Foundation. Many of my postdocs are supported by Komen fellowships, and we also receive funding support from Komen Foundation for some of the most innovative work that we do in the lab. We also work hand in hand with patients, advocates to lobby for breast cancer research, and also for access of care for metastatic breast cancer patients. And this is some of the one of the pictures I did a lobbying in the Capitol Hill a few years ago with Rachel, a metastatic breast cancer survivor in New Jersey. A photo taken in front of the office of Cory Booker. So just to show you that, again, we are a team, one team working together to try to overcome the challenge of metastatic breast cancer.
My name is Nicole Aiello, and I'm a postdoc in Dr. Kang's lab. My presentation will focus on our lab's work on stem cells in mammary gland development and in breast cancer. And please feel free to ask questions in the chat as we move along. So in the background of this slide, on the right, I have a micrograph of a normal mammary duct. And on the left is a micrograph of a tumor. So in the normal duct, you can see these green cells, which are called luminal cells on the inside of the duct, and red cells, which are called myoepithelial cells on the outside of the duct. And you can see that in the tumor, those same cells are present, but they're completely disorganized. And this time we see the red cells on the inside of these ductal structures and the green cells on the outside. And so one of the strategies that the Kang lab uses to approach breast cancer research is to uncover normal mammary gland biology first, and then see how that's altered in the cancer setting. And the hope is to identify and target vulnerabilities in these cancer cells. So in this diagram, I'm showing you a structure of the general structure of the mammary gland in humans and in mice. And so it looks kind of like a tree with a trunk starting at the nipple, and then ducts come off of it. They look like tree branches, and eventually they end in what are called terminal duct lobule units. And if we look at a cross section of one of these ducts here in B, you'll see that there are a few different cell types in the duct. We have luminal cells on the inside, and they're surrounded by what are called myoepithelial cells. And then there are other cell types such as fibroblasts, which act kind of like connective tissue, and they interact with the epithelial cells. And we have adipocytes, which is another name for fat cells. And if we zoom in even closer into the duct, you can see that these luminal and myoepithelial cells actually contain some different cell types, such as alveolar cells, which produce milk, and ductal luminal cells, which kind of form the structure of the gland. And these different cell types are maintained by stem cells. So stem cells are a kind of special cell because they can self-renew, which means that they make copies of themselves, and they can produce differentiated, or you could also say specialized, daughter cells that carry out the function of a tissue. And so this little diagram is kind of a very simplified version of what an embryonic stem cell might behave like. So an embryonic stem cell can produce all of the different cells of the body. And also adults have stem cells in various different tissues, and these adult stem cells maintain those tissues, and they replace old or injured cells to keep the tissue functioning properly. So the mammary gland, of course, has adult stem cells. And here I'm showing you a hierarchy of the different cell types within the epithelial hierarchy in the mammary gland. So on the right, we have our differentiated or our specialized cells. So we have our secretory cells that produce milk, ductal cells for structure, and then we have myoepithelial cells that squeeze the duct and move the milk along. And then in the center of the hierarchy, we have what are called progenitor cells, which are capable of producing maybe one or two of these more specialized cells. And then all the way to the left at the apex of this hierarchy, we have a stem cell. And so these are capable of producing more copies of themselves, but also all of these other more differentiated cell types. And this kind of hierarchy is common in different tissues like the skin or the intestine and of course the mammary gland. But the problem is that tumors often contain cancer cells that behave like stem cells. So at the left, I have a very similar hierarchy with the stem cell on the left and the more differentiated cells on the right. So the problem is that if you have a mutation in a stem cell or even in a more differentiated cell, this can sometimes result in a cancer stem cell. And these cancer stem cells can then go on and produce a tumor and all different kinds of more differentiated but kind of disorganized cell types within that tumor. And in addition to this ability to form tumors, cancer stem cells also tend to be resistant to chemotherapy. And this is because stem cells in general are resistant to DNA damage, and they have to be in order to survive long term and maintain tissue throughout the life of an organism. And cancer stem cells, since they can form primary tumors, they also, unfortunately, are pretty good at forming secondary metastatic tumors. So our lab and others have found that normal and cancer stem cells share molecular similarities. And so if we understand the normal variety, we hope that it will help us tackle the malignant variety. So I'm going to highlight 3 examples of studies from our lab that have uncovered molecular mechanisms of normal and cancer stem cells. These first 2 studies have already been published, and they have to do with the interaction between mammary stem cells and macrophages, which are a type of innate immune cell. And then this last one is going to be about my own ongoing work. So this first study was done by Rumela Chakrabarti, who is a former postdoc in our lab and now has her own lab at UPenn. She found that mammary stem cells produce a high level of a protein called delta-like ligand 1, or DLL1. And then when she deleted that gene in the mouse mammary gland, she found that the ductal tree did not form properly. So you can see here in purple is all the ductal epithelium in a normal mammary gland. And then at the bottom in our knockout mice, that tree is impaired. So it turned out that macrophages express the receptor for delta-like 1, and this receptor is called Notch. And when DLL1 positive mammary stem cells interact with these macrophages, it causes the macrophages to express another protein called Wnt. And Wnt is known for its ability to support stem cells in many different tissues. And so Rumela uncovered this really interesting interaction between stem cells and another type of cell in the microenvironment that supports their function. Now, the second study was done by another postdoc in our lab named Tony Celia Tarrasa, and he also has his own lab now in Spain. Tony identified a microRNA, which is exactly what it sounds like. It's a very small RNA molecule, and he discovered this one called MIR199A, which happens to be very highly expressed in normal mammary stem cells, but also in breast cancer stem cells. He found that when he forced differentiated mammary cells to make this microRNA, they acquired stem cell abilities that they wouldn't normally have. So microRNAs suppress genes, and miR-199 in particular suppresses this gene called L-core. And L-core responds to another protein called interferon alpha. Interferon is made by a bunch of different cell types in the mammary gland and in breast tumors, but in particular, it's secreted by mammary stem cells, cancer stem cells, and macrophages. And when stem cells encounter this interferon, they start to lose their stem cell properties and they become senescent, which means they kind of just sit there and they don't really function anymore. But stem cells can avoid this from happening by making this miR-199A and basically rendering themselves unable to respond to this interferon. So this means that miR-199A kind of promotes the cancer stem cell phenotype, and it might be a good molecule to target therapeutically. So now I've showed you two examples of how macrophages are important for regulating mammary stem cells, but my project is focused on other cell types. So what other types are interacting with mammary stem cells to regulate them? So I've been trying some different approaches to label what we call the niche, which is the immediate microenvironment around a mammary stem cell, with fluorescent proteins. And so I do this using a confocal microscope with a laser that can basically, after I identify a mammary stem cell, I can activate that tissue, that area with green fluorescent protein using my microscope. And then I break the tissue apart, I collect those green cells, and then I can use this technique called single cell RNA sequencing to look at the gene expression in those cells and identify their cell type. So using this technique, I've been able to identify a few different cell types that exist in close proximity to mammary stem cells. And some of these cell types include fibroblasts, which has that connective tissue type cell. Of course, I found macrophages. We know they already play a very big role in maintaining mammary stem cells. I found another cell type called luminal progenitor, which is a little bit more restricted than a stem cell. Endothelial cells, those form blood vessels, a couple of muscle cells, and an assortment of other immune cells. And all of these were found right next to mammary stem cells. And so I've used this information to do some pretty simple co-culture experiments where I mix mammary stem cells with different niche cell types in a dish to see if any of them can enhance the stem cell's ability to self-renew. And so far I've gotten some positive results, in particular from these endothelial cells that form blood vessels, which is kind of interesting because in my system, these endothelial cells are not able to form blood vessels, but they're still able to somehow interact with the mammary stem cells and promote their self-renewal. And I've also found this to be true for cancer stem cells. So the plan is next to investigate the cancer stem cell niche and potentially the metastatic stem cell niche. So the next logical step of this research is to determine the signals in the niche that support stem cells and then try to target those interactions therapeutically to break down the support surrounding cancer stem cells. And if we can do that, then we'll have another strategy to fight breast cancer and metastasis. So I'd like to thank the Komen Foundation and everyone watching for your support for our research. And now I will turn it over to Mark, who's going to discuss his translational research.
Hello, and thank you to the great introduction from Dr. Kang and from Nicole for explaining how our basic research is being translated into new and innovative therapies for the breast cancer patient. And so today I'm going to talk about taking some of those basic discoveries from the lab and trying to translate those into new therapies, how we're doing that, and what it looks like when we're moving those towards clinical practice to help the patients and their families. And so my story, I think, like many other people, starts when I got into research. There was a diagnosis of metastatic cancer in my family. And that was something that made me quickly realize that there are not great therapies that have been developed for this entire patient population. And it is a very critically critically important process that we start to research novel mechanisms and translate those to patients so that everybody can benefit, both the patients and the families. And so to give you a quick background on myself, so I originally completed my PhD at Princeton University under Dr. Kang with a focus in molecular biology of cancer. And since joining the lab at Princeton, I've accumulated more than 9 years of experience, both in this discovery as well as focusing on druggable targets and then translating those into therapies that I'll give you a bit of a preview on today. And my specialty is really in that area of biochemistry, and we mostly focus on enzymes and how those enzymes can be targeted. Historically, enzymes are the most successful class of biological targets for moving therapies from basic research into the clinic. And with some of that research that we've done, we've founded, together, Dr. Kang, myself, a new company, and have spun that out of the university and are now using that company as a vehicle to generate new therapies to try to help the metastatic breast cancer patient. And so our mission overall is to really change the survival paradigm for patients with advanced cancers, not only in breast cancer, but in solid tumors across the entire spectrum. And so in particular, when you look at some of the most major cancer types that are diagnosed in the US today, you find that many of those patients are newly diagnosed with advanced cancers. Those are stage 3, stage 3B, stage 4 cancers that have already spread to regional sites beyond the primary tumor site. And this becomes a really critical need when you look at the 5-year survival metrics for those patients that have been diagnosed with localized cancer versus those that have been diagnosed with advanced metastatic or immunosuppressive cancer. And of the patients that will unfortunately die from cancer in the coming year in the US, the vast majority of those will die because there are no effective therapies for these late stage and advanced cancer patients. And so our mission is to develop these new therapies that will work on the advanced cancer patient and will in parallel work on those tumors in the primary tumor situation. And so I'd like to focus in on 2 different projects that we're working on. One of which is a Komen funded work, which explores E-SELECTIN as a novel target in metastatic breast cancer. This study was published in Nature Cell Biology about a year ago. And what I'll start out with is a broad summary of what we found, which is when you think about cancer metastasis, you realize that there are really 3 essential components. There is the primary tumor where the tumor started out and is growing. And then you have that tumor cell as it circulates throughout the body during cancer metastasis, and the components of the host system or the stroma that Dr. Kang mentioned that tumor cell will interact with on its journey. And so here we have a diagram of a cancer cell on the left and the bone marrow capillary that those cancer cells interact with during bone metastasis. And so what we found was this novel protein that's called GLGI that has a modification at one of its sites that actually adds these unique types of sugars to that protein. And that is done by enzymes that are named FUT3 and FUT6. They're unique fucosal transferases. And once those, which are specifically expressed in metastatic breast cancer cells, when those are expressed and they're active, they modify this GLGI protein, which makes the GLGI go to the surface of the cancer cell. At that point, it can actually physically interact with the blood vessels that are exclusively found in the bone marrow that express this protein called E-SELECTIN. And once this GLGI found on the cancer cells binds to E-SELECTIN, what happens is it goes through this molecular signaling cascade that makes the tumor cells change their shape and actually become more competent to then grow inside the bone. And so theoretically, blocking one of the steps of this presentation of GLGI on the surface, or its modification, or its binding to E-SELECTIN, should theoretically offer new therapeutic node in the treatment of metastatic breast cancer. And so I'll give a real life example of what this looks like during bone metastasis. So in this image, you can see in the purple is the expression of GLGI, and green are the breast cancer cells themselves that are growing in the bone. And in that light cyan color, that's the E-SELECTIN protein that is expressed on those blood vessels. And what you can see is that these three different components are all mixing together, and that allows the cancer cells to grow in the bone marrow. And so going into how we discovered this, what we started out with was this decades-old hypothesis of how E-SELECTIN works. And what we found is that in normal adults and in the mice that we work with, E-SELECTIN is expressed exclusively in the bone marrow. And what you see right here in the green is E-SELECTIN, and in the red is the actual blood vessels that grow within normal bone marrow. And when we generated mice that no longer had the ability to express this E-SELECTIN protein, what we found was that breast cancer cells could no longer metastasize to the bone in these mice, which validates that hypothesis. And going more and more specifically into the mechanism is we looked at one of these breast cancer cells that metastasized to the bone. And in red, we were looking at the expression of this GLG1 protein. And in green, we were looking at the ability of the E-SELECTIN protein to bind to GLG1. And what we showed is that GLG1 is the main binding partner to E-SELECTIN. So if you think about this a bit like Velcro, the cancer cells are expressing one half of the Velcro, and then bone marrow always has the sort of sticky half of the other side of the Velcro. And for those cancer cells that express GLG1, they can stick together. And by sticking together, it creates this vicious cycle that allows the cancer cells to grow within the bone. And so what we did was we actually partnered with an emerging innovative biotech startup called GlycoMimetics. And we used one of their drugs, which we call GMI 1271. It's called UPROLESELAN in the clinic right now. And we showed that treatment of mice that had been injected with these human breast cancer bone metastatic cells could actually alleviate the progression of bone metastasis. And once this study was published, it provided the rationale for moving into the clinic this novel drug from GlycoMimetics that was actually developed to treat sickle cell disease, but we found had this really compelling reason to move into clinical patients to treat bone metastasis. And so this study is currently underway at Duke, and we're anxiously awaiting the results of it. Now, moving on to other projects, and these are the programs that we found had tremendous therapeutic potential and were internally discovered from very basic mechanism in the lab. And during the latter half of my PhD, I translated into new first-in-class therapies that we're now trying to further develop through an iterative process of medicinal chemistry. These we're trying to move into the clinic in the next 3 to 4 years with triple-negative breast cancer as one of our key patient populations that we're seeking to help with these. And our overarching mission here is that we're going to move towards curing these conventionally incurable cancers. So we want to develop therapeutics that work in the late-stage disease where no other or few other therapeutics have been successful. And so I'll go a little bit more into what these two different targets look like. One is a novel immune oncology program that we're trying to bring the benefits of immunotherapy to breast cancer. And the second one is a target that is responsible for handling fat metabolism in the cancer cells. And we've actually found that there's a unique type of fat metabolism that the cancer cells need to process in order to grow and survive and resist the stress that Dr. Kang mentioned was so critical to their survival in the metastatic patient. And so our immunotherapy program is one where we're looking at the dance that happens between the immune system and the tumor cell. And we've known for a very long time that these cell types that are called effector T cells are one of the main reasons why cancer either can't grow in the first place or is eliminated. And back in the 1980s and 1990s, early doctors discovered this therapy that's called Proleukin, which can enhance the ability of these T cells to attack the tumor cells. However, the tumor cells can fight back through pathways that are called PD-1 or CTLA-4. And one of the most famous drugs for blocking this tumor's defense mechanism is Keytruda, which has been tremendously successful for patients with melanoma and melanoma and lung cancers. However, what we also find is that the tumor cells are able to generate this other immune suppressive cell type, which is called a regulatory T cell. And these regulatory T cells are actually dominant over the effector T cells. So if the tumor cell can help to generate these regulatory T cells, even Keytruda often won't work in those patients. And so what we were looking for specifically was a therapy that could block the formation of these regulatory T cells. And we traced this back to another cell type, which is called a regulatory dendritic cell or tumor-associated macrophage. And we're currently developing therapies that block this signaling pathway that generates the regulatory T cells. By doing this, we hope to create an immune permissive environment in the primary and metastatic tumors that allows normal immunotherapy or the patient's own immune system to eradicate the tumor cell and thereby offer durable and long-lasting remissions for those patients. The second target that we're pursuing is that one that is involved in a unique type of fat metabolism. And this fat metabolism is important when that cancer cell is undergoing things like chemotherapy or being exposed to the high oxygen environment in the blood. And those stresses lead to changes, molecular changes in the fats within that tumor cell. And we found a unique enzyme that is upregulated in these very aggressive tumor cells that allows them to handle that fat metabolism and to grow even in the face of many, many different stressors that the tumors often go through during clinical treatment, such as chemotherapy or other targeted therapies. And so this is a therapy that we've already shown it synergizes very well with other standards of care like paclitaxel, and is actually even effective in treating established metastatic disease in breast cancer models and other solid tumor models. Now, we already have a therapy here that is very close to entering into the clinic. We're pursuing final touches and a few molecular changes to the molecule to actually make this an orally available therapy that we can hope have an outsized effect on these late-stage breast cancer patients. And so to give you an idea of what this looks like, when we look in our mouse models with this target, what we see is that when we take metastatic breast cancer cells and we knock out this enzyme, the cells can no longer metastasize in the mouse. However, when we rescue it with that enzyme, so we add that enzyme back in, it restores the ability of these breast cancer cells to metastasize. And then finally, if we restore it with a defective type of protein that is no longer able to metabolize those unique fats, again, those cells can no longer metastasize, and this would mimic the same thing as a drug therapy would. And so the true benefit to patients here is that when we look at triple-negative breast cancer patients, many of which express this target, those that are diagnosed with an early-stage disease where the primary tumor is localized tend to survive for more than 5 years and perform very well after the current standard of care treatment. However, for those patients that are diagnosed with late-stage and metastatic disease, fewer than 15% will survive out to 5 years. Now, this target that we're pursuing is highly active in all of the different triple-negative breast cancer models that we've tested so far, and our therapy is highly effective in reducing the enzyme activity in these different breast cancer models. And when we use these therapies on the human breast cancer models that we've introduced into mice to form metastases, we can actually lead to the almost complete stasis of those metastases and extended survival in these mouse models. And so this team that is developing these therapies and pushing them towards the clinic is myself and Dr. Kang, who is working as a close advisor with us. We've also hired several medicinal chemists that have a lot of experience from the pharmaceutical area, coming from Boehringer and Merck and other major biotechs, as well as an advisory team that is helping us through this very difficult drug development process and hopefully transitioning these therapies from basic research that was funded in part from Susan Komen into novel therapies that can hopefully help metastatic cancer patients in the coming years. And finally, I just really want to highlight that we are pursuing this novel paradigm is that therapies to treat and reverse advanced and immunosuppressive cancers are critically needed. Those patients that are diagnosed with localized cancer are treated quite well in the current clinical practice, while those that are diagnosed with advanced cancer are in critical need. And those therapies to reverse the cancer and metastasis in those patients are ones that we're focusing on, and we're trying to discover those therapies. And with that, I'd like to thank you all for your support and Dr. Kang for his mentorship and providing all the help that we need in pursuing these incredibly important and meaningful research goals. Thank you.